Hello students. Today I, Shweta Singh, bring to you another lecture on the topic the fundamental unit of life of class 9th biology. In this lecture we are going to discuss about cytoplasm and the cell organelles present in the cytoplasm of a cell. The cytoplasm is a thick solution that is filled inside the cell and is enclosed by the cell membrane. The cytoplasmic content of the eukaryotic cell is different from a prokaryotic cell. As you can see in this diagram, the eukaryotic cell is somewhat larger in size as compared to the prokaryotic cell. The eukaryotic cell also contains a central, well-defined nucleus, whereas the central portion in the prokaryote is not well defined and is thus known as the nucleoid instead of nucleus. The eukaryotic cell contains certain membrane-bound organelles such as the mitochondria, which are not visible in the prokaryotes. Some functions of the cytoplasm. It supports and suspends the cell organelles within the cell. All the cellular processes occur in the cytoplasm, including the formation of proteins. It allows movements of substances in the cell, such as the various hormones. It dissolves the cellular wastes. Now we are going to talk about the cell organelles of the eukaryotic cell in detail. These eukaryotic cells contain many specialized cell organelles. Each of these cell organelles perform a specific function for the cell. These cell organelles keep the various chemical activities of the cell separate from the other ongoing activities of the cell. Because these organelles are enclosed by the plasma membrane, therefore they are known as the membrane-bound organelles. A very interesting fact about the plasma membrane that we are talking about here is that the viruses are not considered as living because they lack these membranes, and hence they do not show the characteristics of life until they enter a living body and use its cell machinery to multiply. So by now we know that a cell consists of three features: the cell or plasma membrane, the cytoplasm and the nucleus. The cytoplasm contains a cytosol, that is the liquid part, and many cell organelles inside this liquid part. These cell organelles can be of two types: non-membrane bound cell organelles and the membrane bound cell organelles. Again, these membrane bound cell organelles can be of two types: single membrane bound cell organelles and double membrane bound cell organelles. Single membrane bound cell organelles means there is just one layer of the cell membrane on the organelle, whereas the double membrane bound cell organelles means that there is double layer, one inner and one outer, over the organelle. So which are the single membrane bound and which are the double membrane bound? The single membrane bound cell organelles are the Golgi apparatus, endoplasmic reticulum, lysosomes and vacuoles. The double membrane bound cell organelles are mitochondria and chloroplast. And students, don't forget the nucleus also is a double membrane bound structure. Okay, so now let us write down some differences between the prokaryotic cell and the eukaryotic cell. But here are certain blanks. Let's fill them up. The prokaryotic cell is generally small in size, whereas the eukaryotic cell is larger in size. In the prokaryotic cell, the nuclear region is not well defined. It does not contain the nuclear membrane, and hence it is known as a nucleoid and not a nucleus. Whereas in the eukaryotic cell, the nuclear region is well defined, known as the nucleus, which is surrounded by a double layered nuclear membrane. In prokaryotic cell, only a single chromosome is present, whereas in eukaryotic cell, more than one chromosome is present. If you may remember, humans contain 46 chromosomes. The membrane-bound cell organelles are absent in prokaryotic cell, whereas these membrane-bound cell organelles such as mitochondria, chloroplast, Golgi complex are present in the eukaryotic cell. The first organelle that we are going to discuss is the endoplasmic reticulum. Endoplasmic means inside the cytoplasm. Reticulum means a network, an intense network. Therefore, the endoplasmic reticulum is a large network of membrane-bound tubes and sheets. It looks like long tubes or round or oblong bags, as you can see in the diagram. It is the largest organelle and extends throughout the entire cytoplasm. Within a cell, there are two types of endoplasmic reticulum, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Have a look at this diagram. This portion is the rough endoplasmic reticulum because it appears rough due to the presence of ribosomes on the surface of the reticulum, whereas this lighter portion is the smooth endoplasmic reticulum, which does not contain any ribosomes, so it appears smooth. So what does this endoplasmic reticulum do within the cell? The rough ER contains ribosomes, and these ribosomes are the site for protein synthesis. That is, all the protein which is made inside the cell is made on the rough ER, whereas the smooth ER is associated with manufacturing of fats or lipids. Most of the proteins and fats are used up in making cell membrane, and this process of making cell membrane is known as the membrane biogenesis. So we can say that the endoplasmic reticulum is also responsible for membrane biogenesis. Now, some of the other functions of endoplasmic reticulum. Transportation of materials between the different parts of the cytoplasm due to its extensive spreading throughout the cytoplasm, and also between the nucleus and the cytoplasm. It also acts as a cytoplasmic framework and provides a surface for some of the biochemical activities of the cell. Apart from synthesizing proteins, the RER is also responsible for the proper folding of proteins to make them functional. The liver cells of the vertebrates contain SER, which are responsible for detoxifying poisons and drugs out of the cell. The next organelle that we are going to discuss is the Golgi apparatus. It derives its name after its discoverer, that is Camillo Golgi. These Golgi apparatus is a collection of parallel stakes of tubules or tube-like structures. These stakes are known as cisterns or cisternae. These vesicles have their own membranes, and these membranes are sometimes connected to those of the ER. Why are these connected to the ER? Because the main function of Golgi is to carry materials synthesized by the ER to different parts of the cell. So basically, it is responsible for the storing and the packaging of the materials synthesized in the ER, materials such as proteins and fats. Also, it is responsible for formation of complex sugars within the cell and the formation of lysosomes. Lysosomes? Yes, lysosomes are the next cell organelles that we are going to discuss. So what are these lysosomes? Well, lysosomes are single membrane vesicles which are responsible for the cleaning of the cell because they can digest any foreign material, food, bacteria, or even the worn out cell organelles. But how can lysosome digest any foreign material that enters the cell? Lysosomes are capable of doing so because they have special digestive enzymes known as the hydrolytic enzymes. These enzymes are specialized to break the materials and digest them. These enzymes are synthesized by the rough endoplasmic reticulum and packaged into lysosome by Golgi bodies. So students, can you see a chain over here? The enzymes are synthesized by the rough endoplasmic reticulum, are sent to the Golgi complex for packaging, and then they are packaged into lysosomes and distributed throughout the cell. Isn't it amazing? Another amazing fact for lysosomes is they are called as suicide bags. Why? Because during the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest the whole cell. Therefore, the lysosomes are known as the suicide bags of a cell as they lead to the death of the cell. Here in this diagram, you can see how the lysosomes are able to digest the foreign food particles. The food particle is entering into the cell. The lysosomes are present in the cytoplasm. They combine with the food particles, digest them into smaller fragments, and the waste material is eliminated out of the cell by exocytosis. The next organelle in our list is mitochondria. Mitochondria is a very special organelle because first, it is a double membrane organelle, and secondly, it contains its own DNA. Till now, we studied that the nucleus contains the DNA and the genetic information is stored in this DNA. But mitochondria has got special privileges to synthesize some of its own proteins. Hence, it has been gifted by a small portion of its own DNA. That is why it is also known as a semi-autonomous organelle. The cell requires energy in order to carry out several activities, and this energy is generated by the mitochondria in the form of adenosine triphosphate, in short ATP, which is the energy currency of the cell. Because the mitochondria synthesizes the energy in the form of ATP, it is also known as the powerhouse of the cell. The mitochondria is also the site for cellular respiration. Cellular respiration means the breakdown of carbohydrates in the presence of oxygen to release energy. Again, energy in the form of ATP. As I said, the mitochondria is a double membrane organelle. Hence, it contains an outer membrane which is porous in nature and the inner membrane which is deeply folded. Here, see this diagram. The inner membrane is deeply folded into the matrix. The matrix is the empty space within the mitochondria. The inner membrane of the mitochondria is known as the cristae. The function of cristae is very essential because it increases the surface area by forming these folds or invaginations, thus increasing the amount of ATP molecules generated by the mitochondria. Okay, so now let's talk about the plastids. Remember, plastids are only present in plant cells. You do not find plastids in the animal cells. In the plant cells, plastids are of two types, chromoplasts and leucoplasts. The chromoplasts are the colored plastids and leucoplasts are the colorless plastids. It's simple to remember, chromo, colored, leuco, colorless. The color of the chromoplast is due to the presence of some pigments. Like for example, if the green pigment chlorophyll is present, then the plastid is given the name as chloroplast. Yes, chloroplast. Does it ring a bell? Yes, of course you're right. It is important for photosynthesis in plants. But apart from chlorophyll, the chloroplast also contains certain yellow and orange pigments. The leucoplasts, on the other hand, are responsible for storing materials such as starch, oils, or protein granules. A very important feature for plastids is, just like mitochondria, it is also a double membrane organelle with an outer membrane and an inner membrane. It also contains its own DNA and ribosome to manufacture its own proteins. Hence, it is semi-autonomous. Have a look at the diagram of the chloroplast. The green color of the plants is due to the chlorophyll present in the chloroplast. These chlorophyll pigments are present in structures like thylakoids, which are stacked together to form a granum, and a collection of these granum is known as a grana. Now we come to the last organelle, the vacuoles. Vacuoles are the storage sacs for solid or liquid contents. That is, if a cell wants to store anything, solid or liquid, it will do so within a vacuole. Now both plants and animal cells needs to store materials. Hence, these vacuoles are present in both plants and animals. But the plant vacuoles are bigger in size, are more in number than the animal vacuoles. Sometimes these plant vacuoles combine together to form a central huge vacuole, which covers almost the entire cell's area. Vacuoles are of three types: sap vacuoles, contractile vacuoles, and food vacuoles. Let's give a quick glance to each one of them. The sap vacuoles are the vacuoles filled with a special liquid known as the cell sap. The sap vacuoles are covered by a semi-permeable membrane called tonoplast, which separates the cell sap from the cytoplasm. These sap vacuoles are responsible for providing turgidity and rigidity to the cell via osmosis. Here is a diagram of the sap vacuole. Contractile vacuoles. They are found in protistans and algal cells in fresh water. The membrane of the contractile vacuoles is highly extensible and collapses easily. That is, it readily contracts and relaxes, hence its name. These vacuoles are responsible for osmoregulation. Osmoregulation means maintaining the water content of the cell and thus are responsible for the excretion in the cells. Have a look at this diagram of amoeba. The water enters the amoeba by osmosis and collects within the contractile vacuole. Now the contractile vacuole will gain water and will swell up. The contractile vacuole bursts to release the water. The process starts all over again. So thus, while releasing the water, it will eliminate out the waste material also and thus perform excretion. The last type of vacuoles are the food vacuoles. They are found in the cells of protozoans and several lower animals. Food vacuoles are responsible for digestion of food as the name suggests because they contain the food digesting enzymes. The digested food then passes into the cytoplasm found in single-celled organisms like the amoeba again. Here, have a look at this diagram. The food particle enters the cell through the plasma membrane. It is combined with the lysosome. Please remember, the lysosome contains these digestive enzymes which will break down the food. What will form will be a food vacuole, this one, which is formed by the combination of lysosome and the food vacuole. The food is digested and ready to release energy. So that is all about the cell cytoplasm and the various cell organelles present in the cell cytoplasm of the eukaryote. I hope you have understood all the major points, all the important points. Please note them down. Thank you.